in the Department of Biology. But I do want to convey and show you some very exciting things that what I have observed along the way in terms of cross-disciplinary types of interests, and that is music. And many of you have thought that what does music have to do with science? And hopefully today I can convey that there is a lot of connection between science and music, and especially to do with biology. So when we talk about aesthetics, and as Ralph had mentioned before, that I am a structural biologist, and what that really means is that I study things at the molecular level. And so what you see in the organismal level, meaning the animals, the shapes, the plants, and everything, and if you go down and get smaller and smaller, you get down to the cells, and when you get down to the cells, you get down to the components that make up the cells. These are the macromolecules that Dr. Gobler was referring to. And so I study a fraction of that, and these are proteins. And so I'm going to talk about proteins. I'm also going to talk about things that you may have heard of, and that is the genetic material and DNA. And I'm going to try to reveal some interest in terms of patterns of DNA and proteins that are related to music and how it may be very aesthetic and why we are intrinsically attracted to it. All right, so I'm going to talk about genome analysis, and so I'm going to try to give a brief introduction about what is genome, what is DNA, for those of you who are not familiar with it, and I'll talk about things that we find out in structural biology and a little bit about health relationships, things like this. Okay, so let me give you the 30-second biology lesson. All right, if you go down and look at a plant or any type of things that are living and just reduce yourself down, you get down to the cell. And this is a schematic diagram of a cell. So anyone who's taken freshman biology would know this quite well. I'm just going to give you a fresh review. If you go inside the cell, this is what you'll find inside. It's like a sack of a bunch of organelles, and these organelles have specific functions. And we're diving into what is called the nucleolus, and that contains the genetic material. And that genetic material is composed of DNA, deoxynucleic acids. And these deoxynucleic acids are really comprised of letter codes. And there are about 3 billion of these letter codes. 3 billion contributed by your mother, and the other 3 billion contributed by your father. And this is what DNA looks like. And so it's coiled up and wrapped up. Each cell that you see in our body is contains 6 billion letters. And all of us, in humans, we have about 1 trillion of these cells among us. So if you spread out all the DNA in end to end, it is said that the length is from the Earth to the Moon several times and back. So you got a lot of information and letter codes in there. All right, so what about deoxynucleic acid that is so special? Because this is what contains the genetic material that composes who we are. And so what DNA is made of nucleotides, and there are 4 chemical compositions, and these are shown by letters, and they're G, C's, A's, and T's. All right, and just to show you what they represent, these are called nucleotides. So knowing the exact compositions of these atoms is not important for our discussion, but I just want to show you that these aren't just letters that are mended together for genetic material, but they actually represent chemical compounds. And so these chemical compounds, as I said, are represented by 4 letters: G, C's, A's, and T's. And any of these combinations can code for specific colors of your eyes, your hair, how tall you are, what is your immune system. Everything about you is comprised of information from this. So that is your 1-minute biology lesson, and I just gave you about 2 years worth of biology in a nutshell. A cell, the composition of a cell, what's inside the cell, the genetic material, and what the genetic material is made out of. All right, now having that said, I want to talk about the letter codes because there's some interesting things that are composed of these letter codes. And I'm just flashing you kind of a snapshot of all these letter codes and to show you what these may look like. So again, as I said, we have 3 billion. That's 3 times 10 to the 9th. And letters of genetic codes that's in our body. And so this is what they look like for a bioinformatician, for a person that is working on a computer to dissect all these sequences and find out what they mean and how these things regulate your body. That is the hard part in biology at the moment. To give you an idea how big this is, 3.2 billion bases, all right? So there are almost 1 million letters in the Moby Dick. So everybody's read Moby Dick? All right, you guys are familiar with Moby Dick? All right, so it's a pretty thick book. Some high schools make you read it, and in college there are a lot of literature classes that make you read it. It's not one of your books that you could read over the weekend, but in 3.2 billion letters, there are estimated to be about 3,419 books of this in a library. This is how many letters we're talking about, just to give you a number in perspective, about 3.2 billions. Okay, now, what I want to talk about is this fellow I met when I was a postdoc at training, and this was a meeting in France, and this fellow's name is Susumi Ono. He's actually well known for looking at molecular genes and regulations, and he's a biologist, he's a biochemist. But what people and biologists in particular may not know about him is that he had a very great passion for music. And what he did in the weekend was very fascinating to me. And so I took on and looked at this very carefully. And this is what he did. He said that if you take those letter codes and now assign it to musical notes, all right? So in the most simplistic fashion, if you take the letters of the nucleotide, and again, there are 4 possible letters, C, G's, A's, and T's, all right? And if you take these letters and take portions of our genome, and now put it into the musical treble clef, for example, or any type of clef, does it provide any musical sense? That was his question. And to many people's surprise, that even with a very simple conversion with the algorithm, if you play this actually doesn't sound bad, meaning that there's some musicality to it. Now, with 4 letters, I mean, you could make a lot of easy trends and things like that. But this was a start. And the question is that can you actually take the majority of the genome, or are there genes, sections that provides information for certain blood type, for hair colors, things like that? Can you actually transpose this to musical notes? All right, so that question came to be with him and many of his students and other investigators that have looked at this very carefully. So I want to provide you a few examples of something that provide some musicality. This is a sequence that I'm showing you in part of an antibody gene from a mouse. Okay, so this gene, this sequences, provides information on proteins that makes antibodies. Does everybody know what an antibody is? All right, so this is the protein that is part of the immune system. It helps you fight off diseases. And an antibody looks something like this. Okay, this is the backbone, the 3-dimensional structures of this. All right, if you actually transpose this into music, and it would sound something like this. And this has the algorithm, specific algorithms that takes different letter codes and put it to certain durations and musical bars. And Lydia, I know you play the piano. You could probably play this. So this piece was written by taking a good portion, about 200 of these nucleotides, transcribed into musical notes through an algorithm based on the notes. The chromaticism is based on whether two letters, consecutive letters, are following each other. Where they are in the scales are retained in segments, so there's certain algorithms to this. It doesn't sound bad, right? And you could almost say that it's actually very, very musical in that it is very consonant. This is what the music looks like if it's written out. And so, of course, when you first transcribe this, it doesn't fall in neatly into its proper clefs. But it starts out with outer range, and I'll show you a little bit of that in the beginning. But usually it's brought down in an octave, or it's brought down to its respective chords. But nonetheless, it provides some pretty good musicality to this. So this is the sequence of the immunoglobulin gene from mouse. And it follows the letter codes for DNA and for proteins as well, too. All right, so what is it about this? Let me talk about the 4 nucleotides. All right, so we have adenine, guanine, thymine, cytosine. This is the A, G, C's, and T's that we're talking about. And here is a possible algorithm, because people write different algorithms for this. And what algorithm mean is that what are the rules that you take from the letters and transcribe it to music? And here is one possibility. This is a common one that is written. All right, you give each letter a numerical value. And in this case, if it's the DNA, you give A0, T1, C2, and so on. And you assign a digital value for each of these letters. The second thing is you determine a pitch value. All right, so this is a fancy formula, but all it says is really you take maximum pitches and you look at maximum, minimum, and trying to fit it with that certain pitch. So that means that if we're restricted to certain octaves, so we don't expand more than 2.5 octaves or an octave or 2 octaves. It really depends. So you set your maximum and minimum values in pitch. Right? So these are the rules that we take by. You establish a duration. You assign a time value for each letter or each letter combination for these. And like I said, these algorithms can change depending on who writes these algorithms or how you want to approach it. Except that particular ones that I showed you follows this particular algorithm for this. And this one you could actually get online, and for anyone who wants it, I'll give it to you. You could actually submit your gene code for this, and it will provide you with a score of music. And it'll be raw material. It's up to you to kind of refine it and things like this, but it actually works out. So this is one example. There are many different types of algorithms that you can apply to this. This is the so if you take you declare a maximum pitch to this. So for example, what I didn't define here is that if you take an octave, and let's say 2.5 octaves is usually in the span of an amino acid code, all right? And you could say that O is the higher octave, and for a higher N and a lower N. And then you have a pitch values for these notes. So this is just an assignment to establish a range or a window of your notes. Okay, so before I go on, I want to talk about the passage of genetic information. And this is called the dogma of molecular biology. It's DNA material. When genes are expressed, it goes through this pathway where it turns into an intermediate molecule called RNA, which is ribose nucleic acid, and then in turn it goes to protein. All right? When your cells are being expressed, that means that it takes your genetic material information, the A, G, C's, and T's, and combinations, and the cell knows that how to convert that into an intermediate template, which is called RNA, and then it makes proteins. And proteins are polymers, or a composite of now further letter codes, and there are 20 letter codes. These are called amino acids, right? So we learned that in DNA there are 4 nucleotides. In proteins there are 20 amino acids, so 20 letter codes. And so one of the questions is that if we look at DNA, let's look at proteins. Right? With protein sequences, you could apply the same thing. You put letter codes to it, you put musical notes to it. Is there any musicality to this? All right, so this is just an example. Again, here is now a second year biochemistry on 30 seconds. All right, so types of proteins that you're maybe familiar with are insulin, different immunobodies, antibodies, as I said before, and any types of these is a product of your DNA coding. And it's made up of these amino acids. There are 20 of them, right? And so this is what they look like. This is, if you take a biochemistry class, this is what you'll have to learn. But for right now, I just want to show you their existence and their chemical compositions and what they look like. But more importantly, I want to show you the code to them. All right, so glycine has one-letter code, G, alanine, A, valine, and so forth. All right, they're assigned one-letter codes. So any amino acids or any proteins, you could have an assignment of all these letter codes, and this will be a signature of that particular protein. Okay, now, if you take the 20 amino acids and now try to assign a note to them, and let's just take the classic keyboard, 88 keys. And 88 keys, there's about 2.5 octaves that you can fill with the 20 amino acids. And again, those of you who do not play the piano, I laid out the keyboard for you, and these are the notes that are corresponding to the keys. And what we're going to do is we're going to lay out the amino acid assignments to this. Right? So each of these letters represents an amino acid. Each of these letters represents a chemical that is part of a protein. And the question is that if you assign these notes and now put them into music and write it out, does it sound musical? Right? So we're going to have to apply a little bit of rules to it, and some of the things may be like this. All right? So if you take a certain number of amino acids, and this is our perspective, if you take, let's say, these compositions, W, P, M, H, right? Again, they correspond to chemical compositions, and let's assign them to these notes here. All right? Now, let's take the second set, and let's assign them to these. Now, some of these may have inversions in these chords, all right? And so this is part of the algorithm, is that, well, these particular amino acids, depending on their frequencies that you observe in nature, some of them will be assigned in first inversions or in root positions, all right? In root positions, meaning that they are assigned every other note, they're stacked on top of each other, or there are in inversions where they are separated out. And again, here's some examples of the note assignment. All right, so this is an example where you take your 20 amino acid, your chemical composition, and you assign them specific chords to these. Now, I taught a bioinformatics class last year, and I had students write a program that actually translates this and trying to give out some musicality to this. Now, the raw material looks something like this. I'm going to skip this one. It looks something like this. All right, so those of you who are musicians out there, you probably can read this, and you could probably hear in your head it's going to sound all over the place. All right, if you just write the program, this is what it's going to give you. And sometimes it's even worse. It's not even, it's far off range, and they're not even close. But if you write it in a way that if you bring it down within a certain octave, so let's say your range has to be within 2.5 octaves. So the rule of thumb is that if your note is corresponding to, let's say, 3 octaves away, you bring it down 2 octaves. If it's the same thing lower, you bring it back up. So if you bring everything back up, then you would have the combinations of these notes, and they tie together, all right? And so you would have something like this. Now, if you play this, it actually sounds rather musical, Right? There is a continuous stepwise flow in many of these, and the chords, in most cases, are consonant to each other. All right, now, these are just examples, and this is why I was showing you some of the algorithms behind it. This is an example of how we convert certain amino acids into music for this. I'm going to show you an example. This is a protein called alcohol dehydrogenase, and it's an important enzyme. It's an important protein. Enzyme is a protein. Does anybody know what alcohol dehydrogenase does? Right? Your body uses it to break down alcohol. Right? It breaks down alcohol, so this is the enzyme that everybody uses that breaks down when you're highly toxic or you're drinking alcohol. All right, the amino acid sequence for this is laid out here. These are one-letter code. Remember, each of these letters corresponds to a chemical. And so in a protein, you're made up of any of these 20 amino acids. And if we assign these values to a melodic scheme, and the percussion is just put in just for effect, but the melody is played after this. And this is played directly. You know, again, as I said, the transposition, if it goes off key, is brought back down to a nearby octave. And this is how it sounds. You may not say, well, it's not Mozart, but it doesn't sound bad, all right? And so the question is, why? Why doesn't it sound horrible? However we do this, if we put these in certain combinations of these and to the rules that we applied, none of those really sound so bad that you have to run away from it. All right, and I'll address that a little bit later. Okay, now, right now I said if you take certain DNA sequences and you transpose it to musical notes, you get a certain musical score and you play it and it has certain musicality. If you take the same thing with proteins made of 20 amino acids and transpose it to musical notes, and does it sound musical? And my argument says, for the most cases, it does. Now, what about the other way around? What if you take known music, right, and now transpose it back to DNA, and take that DNA sequence and put it through a public gene bank and find out if there's any genes that matches it. And this is what and this is not my work, this is actually published work, and I thought it was just amazing. So here's a piece. Lydia, you know this one. All right. So this is a famous Chopin piece, Nocturne. Everybody knows it. Many people or pianists have played it. Okay, now, the rules are reversed. Let's take those notes and now transpose it back to DNA code. And what will you get? All right. It turns out in this particular case, that is the letter code that you will get from Chopin's piece here. All right. Now, you could take these sequences and now run it through a bank. And this is a public bank which has all the genome sequences available. And you're asking it, what is the match to it? And it turns out for this particular case, there is a match about 95% for mouse RNA polymerase. All right? This is a protein that is responsible for expressing genes in mouse, from DNA to RNA. All right? But the fact is that there are some resounding patterns that gave rise to a sequence that gave rise to a match. And this is just the amazing part of it, or is it not amazing? And that's the question I want to address shortly to come. All right, so now we've converted, there's your mouse. All right, so now we've converted letter codes that we find in nature, and from both DNA sequences to amino acids, and we've converted to music. We took music and we converted back, and we said there seems to be some association with musicality and some connection between the codes and the music. And the question is why? Is this something that is magical, divine, or there's some scientific basis to this? All right? Why are we attracted to listening to some of these? So when I talk about the relationship in terms of how we look at this in terms of structural biology, and what structural biology really is trying to decipher structures at the atomic level, all right, at the molecular level, and that is to look at sizes and shapes. Now, I'm not going to talk about the techniques about getting there, but I want to show you some things that are very prevalent in terms of structures. And I'm going to talk about symmetry. Right? And so what symmetry is that if you take an object and you impose a orientation turn, either 180 degrees, twofold, or a third of a circle, or a sixth of a circle, all right, around a certain axis, and if it gets to one place and it superimposes itself, it has a symmetry relationship. Right? This is called symmetry. So if you could have a 180-degree turn twofold symmetry, a triangular symmetry, a cubic symmetry, and so forth. You can even have mirror symmetry, such as your left hand, your right hand with each other. And you basically, if you're in front of a mirror, that's what it will look like. The imposing movement is a mirror, and you have a superimposition. So in a lot of the molecules that we look at, we have symmetry. This is actually hemoglobin. This is a protein that carries oxygen in your blood. And here, you could see there is symmetry. That means that you can move this 1/2, 180 degrees, or a quarter of a circle, and it's superimposed onto itself. These are different proteins. These are membrane proteins. They're receptors in your cells that are responsible for uptaking certain molecules. They have beautiful symmetry. Now, this is not made up. This is not some computer animation. This is based on experimental data that gives rise to having definitive positions of the atoms and placed in, and they're tracking the outlines of this. And what I'm showing you here is not aesthetic. It's not only aesthetically pleasing, but has biological function that is required to have these types of symmetry. All right. Here's a viruses. This happens to be a tobacco mosaic virus, a satellite one, and this has all sorts of symmetry, things that are related in one unit. So you could see the symmetry here. You could see the symmetry up here. So that means that if you rotate this thing, it will superimpose onto itself. This is what the structure of the virus looks like. All right, this is another virus. Again, look at the symmetry. The symmetry is a repeating unit which has an orientation change and it superimposes onto itself. This is another protein. So I'm showing you different types of proteins that we observe in structural biology that has a really unique symmetry elements behind it. And there's biological necessity to have symmetry. So why am I bringing up this up? So in biology, it's not only the molecular level. You see symmetry all the time. So let me talk about symmetry further and everyday experience. As I pointed out, mirror symmetry is prevalent. Right? You see these in any type of bilateral animals that you see in organisms, in leaves, in faces, in butterflies. Radial symmetry. All right? So a lot of sea life creatures, honeycombs, have radial symmetry. Right? This is actually a six-fold symmetry, so if you rotate this 1/6, it superimposes on itself. In nature, you have some very, very natural symmetry. This is called translational symmetry. It means that if you take one section and translate it and glide it along, you have a superimposition, meaning that it will look identical to what you started out with in trees and snakes and so forth. All right? There are also mathematical relationships. You've heard about the golden ratio in nature, right? This is the proportion number, the golden number, and that is that if you take the distance of the wider distance and divide it by the inner, you'll have a certain ratio. And that ends up to be about 1.6, and that ratio seems to be prevalent in many different places, including the length of your arm, in proportion to your elbows, your fingers in proportion to the joints, all right? And this number happens to be consistent in nature that you see in seashells and in the spirals of the universe. So there's some astounding symmetric and some proportion relationships in nature for this. All right, now I'm going to start going back to the sequence, to DNA. We also find symmetry in DNA sequences. And the very first thing is that these type of symmetries are called palindromes. And the palindromes are things that are read in one direction, but if you go back and read it, it'll be exactly the same thing. So, symmetry by letters. Try it. This is race car, all right? Read it backwards. It'll be exactly the same thing. All right? Taco cat, all right? And so on. These are actually sentences that are palindromes. DNA in our genome has exactly that type of palindrome sequence. Here's a palindrome by words. Fall leaves as soon as leaves fall. All right? If you read that backwards by words, it's exactly the same thing. So why do we find this in a DNA sequence? Well, it just so happens that these kind of palindromes makes the DNA fold upon itself to this kind of a crucifix type of structure. And this is actually pretty important in terms of gene regulation and how genes express themselves. All right. If you look at things that are symmetrically related, and you look at chromosomes, and so and these bars, this block tells you high regions of symmetry, right? This is just a bar graph showing a chromosome with high regions of symmetry related. In our chromosome, the 23 pairs, including the X and Y, all right, these regions that are highlighted in these sequences here are highly symmetric. That means they either have a lot of repeating sequences, they have things that are read forward and backward, they have bilateral symmetry, they have lots of repetitions behind these, and these are very, very heavy. So in DNA sequences, and also in protein sequences, there are a lot of repeating patterns to this. All right, going back to music. So the question I ask is, why do we, when we transpose musical letters from DNA sequences and protein, why do we get some musicality? And here's a reason, and I don't think there's anything magical about it. If we think about all these symmetry elements that are found in nature, especially in sequence codes and things like that, we also find them in music. Here's some example. Translational symmetry. Remember what translational symmetry are, all right? So you find these, such as in Beethoven, you see lateral symmetry changes on these. Bach has a lot, especially in these fugues, that has translational symmetry. And reflection, remember reflection symmetries, mirror symmetries as well too. Chopin waltz has these types of reflection inversion, glide reflections. These are symmetry elements that are written in music and that we are very attracted to. How about temporal reflections? These are symmetry with vertical mirrors, right? Those of you who are taking theory, we're doing a lot of compositions with these types. And so Bach has a lot of these that has symmetry elements that has rise and falls to these. Translational, rotational, right? Mozart loves these. There's a lot of rotational, translational movements in these music that exists already, and they both simultaneously exist in what we find in nature as well as in our music. How about Beethoven, the Fifth Symphony? Everybody knows this. Turns out that there is a proportion in the measures that is associated with the golden rule, with the golden number. All right, and this so happens that in his repeats and his measures, now whether he intended to write it like this or not, it just so happens that his measure of phrases for 372 measures to 228 happens to fall right in that golden ratio to this. So all these numbers have some very uncanny relationship to this in terms of looking at existing symmetry that exists in biology already, but it's also written down in musical elements. This is why I think that it's not surprising if you transpose the sequence codes to these proteins and patterns into musical notes and the other way around, that the probability of listening to something symmetric would be very high. And this is why I think that things would sound musical when you transpose this. Now, what I'm going to show you is a musical fugue that we actually wrote. So at first, I want to show you this chart. You see these letters stretched out? All right, this is DNA. DNA has 4 letters, A, G, C's, and T's. All right? The higher the letters means the more abundant they are in terms of their sequences. And what I want you to notice is that they are clustered. They have repeats, and each time there's a repeat, something further along the line, it also mimics that repeat. All right? And it repeats, and it mimics a repeat, especially right here. You have a pattern, and then later on, repeat, repeat, and then later on, you go through variations, and then that repeat comes back. Does everybody know what a fugue is? All right, so Bach writes a lot of these fugues. And what fugue is that it takes a musical idea, and then it repeats the heck out of it, right? It brings it up, and then you have a response, and then another response, you have all in different voices, right? So I'll give you an example of this, and this happens to be my music class right now that I'm taking with Dr. Borel, and we just recently wrote a fugue.
Is this the real life? Is this just fantasy? Caught in a landslide, no escape from reality.
So this is based on Bohemian Rhapsody. So this is the theme. It's called the subject in the fugue. All right. Now I want you to listen to the repeat response element. And there's links to these. Now the third voice is coming in. It's going to do the same thing. So listen to these repeats. It comes over and over again. All right, I don't know what grade we'll get out of that, but this, going back to the sequence, you have an idea, you have a repeating element that repeats. And in a lot of the DNA, it also kind of have a response to that. You know, it has that same repeating elements to here, and then it goes through some variations, right, that doesn't have any repeating elements, and then it comes back to have these patterns come back. And it made me think about fugues, because we were thinking about fugues a lot, and we were looking at these DNA sequences, and these show up too. Now, why are these attractive? Because they have these same repeating elements that I just played you. Okay, so answer, at least we think that the reason why things are musical is because of the intrinsic symmetry elements that are written into it. And these intrinsic elements are found in nature already. And chances are, if you transpose those symmetry-related information into music, it will sound musical, and the other way around as well, too. What about music and human health? I just want to touch upon about this. And I don't know if anybody's seen Monty Python, The Life of Brian. And remember they were in a Colosseum, they're complaining about the Romans. What have the Romans ever done for us? And so we could also ask, what has music ever done for us? And so this is a pitch for the music department. I gave this talk originally to sell music. So I just want to bring out some pluses for music under this. In terms of health, there's really well-known a lot of studies that physically music can help in physical therapy, from easing pains to motivating people. As you know, the motivation really is real. It increases performances, endurances, helps people eat less, perhaps, enhances blood vessel functions, and so forth. So it has really real physical health benefits for these. It also has been shown that it helps people recover from surgery. It reduces stress, helps put you in meditative states, helps to ease up symptoms of depressions, elevate moods, and so on. And also it helps cancer patients manage stress and anxiety as well too. And I think the list goes on. There are a lot of health benefits that are being used for health therapy at the moment as well too. And we think the underlying reason is exactly what we've been talking about, is that there's comfort in symmetry, there is comfort in the music that we listen to, that we have our liking for symmetry as well too, and it's an audio version that we see in visual. So these are some of the books that I got information from, and if you're interested, I urge you to actually read it. They're actually very, very good on this. And I just want to make a pitch for an education outreach program that we just started at UAH. This is with the music department. It's called MuSTEM. The idea of it is to use music and to empower STEM training. And we think that the reason is because the training behind both music and STEM requires the same thing. Lydia, what do you think that is? It's a lot of diligence, critical thinking, analytical thinking, and a lot of practice, 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 practice. Right? I've seen you guys in the practice rooms all the time. In STEM area studies, it's the same thing. To look at scores critically, to write music critically, to play critically, analytical thinking, it's the same usage of your brain. And our thought that if you can couple music with STEM, it's a very, very good training. So we started this, and we had a workshop on it last summer. And this is in collaboration with Melody Ng and many of the faculty members. And I just want to show you a short clip of some of the things we came out. So these, this is a workshop where we had high school students that had no idea about music nor STEM. Some knew more than others. And again, we were trying to use music to empower STEM training. This is actually the Shelby Center. And this is the music.
It really surprised me how
I think, so in summary, I guess what I'm trying to tell you today is that there is intrinsic patterns and symmetry that highlights the beauty that we appreciate. And in this particular case, music is one of them. And so the music that we hear has intrinsic symmetry. We could look at these symmetry elements existing in nature and vice versa. We could compose music back and forth with each other. And the music that is in the training that's involved, the thinking that's involved, can also empower a lot of our training in outside of the field of music, particularly in STEM areas. Right, so I think I'll end here if we want to open up for discussions.